Hi, everyone. I'm Anne, and this is Steve. Thank you so much for inviting us to present at the CDG conference this year. I remember attending it way back in 2018, which I can't believe was 4 years ago, and finding this large group of people who really understood what we were going through. And I especially remember how much it meant to be meeting and talking to people who knew exactly what we were going through. Those who attended the 2020 conference may know that we were planning to attend then as well, but our son passed away just a couple weeks prior to that. Dr. Kinoshita was, at the time, was very sweetly dedicated his lecture to our son Emmett. And it's with heavy hearts that we dedicate our talk today to Liam Blake, another little boy who had Pig CDG who passed away late January of this year. He had just turned 4. We're forever thinking of Katie, Russell, and their family and mourning their little boy, who reminded me so very much of Emmett. So we're here today to talk about our son Emmett and the journey that we went through, focused on what we did in the research and treatment space. Our son Emmett was born in April 2016. I think similar to a lot of families here, when he was born, we had no reason to think that anything was seriously wrong. He was in the NICU for 10 days, which at the time felt like the longest week of our lives and week and a half of our lives. But then he was discharged after the doctors didn't find anything on the battery of tests that they ran. He loved eating and hated sleeping, loved being held. So basically reminded us of what we heard the typical newborn stage was like. And honestly, I think at the time, we just wanted to believe the doctors were wrong or overreacting. But then he missed his 3-month milestones and he had seizures and he had trouble eating. And that started our first journey getting a diagnosis. I won't get into that too much here since I want to focus on the what's next part. But I do want to share that we struggled so much to get a real answer. After getting different diagnoses for infantile spasms, Otohara's failure to thrive, we finally got his CDG diagnosis when he was 9 months old through whole exome sequencing. We'd already been through so much by this time that I think we were strangely glad about having a definitive answer, because then that enabled us to say, okay, now we have something concrete to work with. And luckily, one of Steve's college friends happens to have his PhD in molecular and medical pharmacology. So he sat us down, explained what DNA was, explained how genetic mutations happen, because we were really starting from like not knowing anything, and then went over the GPI anchor pathway, which is what you see here. We also started to learn about other families that had started their own research and treatment projects, like Matt Mite's story, which we found pretty early on. And that gave us a great start, but the next step we knew was to figure out the lay of the land. So I'm going to turn the presentation over to Steve to talk about how we thought about the different buckets of treatment options, then close out by sharing what we feel like were some of the lessons learned in the beginning.
So Anne and I, like probably most parents of rare disease children, are unfortunately not scientists. So to try to make up for our lack of scientific knowledge, one of the first things we did was going online and scouring the internet for all of the existing research on Emmett's mutation that we could find. We familiarized ourselves with some of the big names in PIG-A and GPI anchor research, and we tried to think about how we could get in touch with them. Through one person connecting us to another person and a little bit of good fortune, we were able to ultimately get in touch with Dr. Taro Kinoshita and Dr. Yoshiko Murakami from Osaka University, and we heard from earlier this weekend. And they were two of the most published researchers in the GPI anchor space. Dr. Kinoshita and Dr. Murakami graciously agreed to work with us, and they've been collaborating with us ever since on various treatment approaches for PIG-A. So now I'm going to go over the different types of treatment options that we've identified. And we've attempted all of these and gotten to various stages in each of them that I'll take you through on the following slides. The first approach, targeted small molecule supplements, involves selecting specific supplements that might work based on the biological pathway. The second approach, substrate replacement therapy, requires a special compound to be synthesized to bypass the faulty gene. The third approach, a drug repurposing screen, is basically testing numerous drug candidates all at once to see if one works. This is also referred to as a low, medium, or high throughput screening, depending on how many drugs are being tested. And then the final approach, gene therapy, attempts to more fully correct the disorder by replacing the faulty gene entirely. So all of these treatments range pretty widely in terms of time and cost to develop. Gene therapy is by far the most time consuming and most expensive. But in all of these treatments, you still need to go through all of these steps on the bottom of the page, in vitro studies and cell lines, in vivo studies and animal models, and then safety tox studies, ultimately the path towards FDA approval via clinical trials. The first treatment approach we tried was targeted small molecule supplements because, frankly, it was the cheapest and the easiest. This is essentially a form of drug repurposing, which I'm going to revisit when I talk about drug repurposing screens. But in this case, we selected specific supplements with the biological rationale as a treatment for PIG-A. The advantage of this approach is that these are FDA-approved supplements with known safety profiles, and they're relatively easy to access at a reasonable cost. The downside is that this is not a very targeted approach. Dosing is difficult to determine, and you never really know what kinds of unintended side effects you might encounter. So based on the research that my college friend that Anne mentioned had done on PIG-A's role in the GPA anchor pathway, there were two supplements that actually had biological rationale as a PIG-A treatment. The first, N-acetylglucosamine, was the input into the biological reaction involving the PIG-A enzyme. And the second, N-acetylcysteine, had been shown to increase the amount of PIG-A in your body. After consulting with our doctors, as well as some other families who had actually tried to give these very same supplements to their kids, we did try giving each of these supplements to Emmett in gradually increasing doses. Ultimately, we ended up discontinuing them after not finding any improvement either physically or in his EEG, but it is something that we're glad that we at least tried. The next approach that we attempted was substrate replacement therapy. So this was a much more targeted approach where the objective was to provide more of the product that PIG-A was involved in making, this molecule circled here in red. The advantage of this approach is that this bypasses the need for a functional gene entirely, yet it's less costly than gene therapy. A downside, though, is that it does involve making a brand new compound that's not FDA approved. So this does bring added complexities versus drug repurposing approaches. Making a new compound takes time and money, and it brings more uncertainties regarding manufacturing, safety, toxicity, and then regulatory approval. Our work in this area was a collaboration between Dr. Kinoshita and Dr. Peter Seeberger, a carbohydrate chemist who synthesized this compound for us in Germany and then sent it to Dr. Kinoshita's lab in Japan for testing. Dr. Kinoshita initially had some very promising results with this compound in vitro, but then his lab had less successful results in mouse models. With further investment in this compound, we do think that we might be able to improve its chances of success, but for now, we've focused more of our financial resources on gene therapy. The next approach is a drug repurposing screen. You've already heard about a very successful example of this in epalrestat, currently in clinical trials to treat PMN2. The benefits of drug repurposing screens are that you can test multiple drugs all at the same time, in some cases, thousands of drugs, and that the development timeline is faster since these are generally FDA-approved drugs that are already available on the market. So manufacturing, safety, and regulatory hurdles are less of an issue. At this conference 4 years ago, we met Dr. Clement Chow, who also presented at the conference this year. He had already done some pretty exciting fly model work with NGLY1 at the time, and he agreed to help us out by working on PIG-A. So he's actually been able to develop a fly model of PIG-A deficiency, and now he's working on a medium throughput screen to hopefully identify some drug repurposing candidates for PIG-A. And then now the final approach, gene therapy. Gene therapy is sort of the holy grail of treatment approaches because it's the most targeted and it gets to the root cause of the disorder by replacing the faulty gene. And this can provide some pretty significant clinical benefits. The downside of gene therapy is that it's very expensive to manufacture and it can take a long time to develop and to get through clinical trials. When we first started to look into treatment options, gene therapy didn't even seem like a remote possibility. But then when we eventually began talking to rare disease gene therapy companies, most of them told us that gene therapy could indeed be an option for PIG-A. And one of those companies actually connected us to Dr. Catherine Meyer, a leading gene therapy researcher who had worked on one of the very first gene therapies to be approved by the FDA. She agreed to help us, and she synthesized a PIG-A gene therapy vector that she sent to Dr. Kinishida's lab for testing. So we're currently in the process of testing PIG-A gene therapy in mice, and we remain hopeful that it will prove effective and allow us to move forward to the next steps. We would still have a very long path ahead, would have to raise a large amount of funding to continue, but gene therapy proof of concept would be an exciting and important step forward. So that's our journey so far. Now I'll turn it back over to Anne, who will close out by sharing what we think some of the crucial first steps are for anybody looking to embark on a similar research journey.
So looking back, these are the steps we felt were big, important ones to start thinking about once you get the diagnosis, if you want to pursue research or treatment. And I guess I should preface this by saying this applies more to probably to ultra rare diseases when maybe there isn't an established nonprofit or group in place where you're really starting from ground zero. I'll go over each of the points in the following slides, but this is an overview. So understanding what and who exists in terms of research, start thinking about fundraising, and determining whether you're going to start your own nonprofit or foundation or partner with an existing organization, identify appropriate biomarkers to test effectiveness, develop a way to document and track patient families, and then consider having a key central location for key information and resources. And we just felt like getting started on these items will really help as you approach researchers, because it shows at least some plan and some thought into what would happen once you get a researcher on board. So the first one is understand what and who exists in terms of research. This one Steve mentioned already, but you kind of have to become an expert on what's already out there so you know where you need to go from a target goal standpoint. So get familiar with papers, researchers, get a sense of what the patient population looks like. Some of the sites that we use were PubMed to find existing research papers, ClinVar to find people that have been diagnosed. I remember checking it every 3 months to see if we had any new PIG-A patients that had just been diagnosed. And then of course, Google or Bing or whatever search engine you use to find any blogs that someone may have started, articles highlighting different families, websites of existing foundations that might be out there. And then the next two are fundraising focus, because regardless of what route you take, you will need funding, and you'll likely want a nonprofit tied to your fundraising so that the donations can be tax deductible for people, and really to give those donating reassurance that there's some sort of structure behind the donations. A lot could impact whether you decide to start your own nonprofit or partner with an existing one. I've listed some of them here, and they include things like what timeline are you working with, what type of cost or paperwork are you equipped to handle, how flexible would your partner organization be to what you want to accomplish, because sometimes maybe those missions aren't fully aligned. When we were thinking about funding PIG-A CDG research, we found a great partner in CDG Care, and they were extremely helpful in guiding us through the process and making sure we were asking the right questions. The next one is identify appropriate biomarkers to test effectiveness. A biomarker is what you use to figure out if a treatment is effective. And of course, biomarkers will change when you expand from in vitro studies to mouse to human trials, because what you observe will vary. Some of the examples of biomarkers are like metabolic changes, EEG findings, X-ray findings, or even simple things like blood pressure, heart rate could be a biomarker for some disorders. We use GPI anchor protein markers, which are basically proteins that were found to be markedly lower in PIG-A CDG boys. We were able to measure that through a process called flow cytometry, which we were lucky enough to get done through the NIH in July 2017. Those are some of the pictures from when we went to the NIH. And also for anyone that hasn't enrolled in a natural history study, I think it's mentioned in the conference, but that it was super helpful for us to get a really good snapshot of where Emmett was at the time and kind of what to watch out for in the future. And then for a couple of the things we tried, like the supplements that Steve mentioned earlier, we also, of course, just looked at how Emma was doing. Was his focus getting better? Was he able to do more in PT and OT? Kind of things like that. And I think those things will be critical for any probably clinical trials for CDG kids. And the last two items fall more into outreach and tracking. First, you will want a way to document and track patient families. Because when you start to talk about people, when you start to talk to people about treatment, you should be able to answer things like how many patients have this disorder, how similar are the symptoms, what's the life expectancy, why. We decided to go the Google Sheets route, mainly for ease of use and accessibility. There were specific things that we wanted to track for PIGA, and this was the easiest way to have people fill it out. Ultimately, though, we are hoping to have a more robust PIGA CDG patient registry, and Dr. Chow's team is helping us to get started on that. And I know CDG Care also has a patient registry, CDG Connect, that hopefully everyone has submitted information for. It's important as we look at CDGs as a whole to have that type of information also. And the last item, consider having a central location for key information. It took us around a year to have this website created, pigacdg.org, but it's a central place where new patients could find us, current families could find things like known symptoms or research, where we could announce things like our fundraiser. Facebook groups are so nice for families to connect on a day-to-day basis, but I think having a public website was so useful to act as somewhere where we could point different groups to as we worked on the other pieces. And so where are we now? From a research standpoint, as Steve mentioned, we are in the process of testing PIGA gene therapy in mice. We have our fundraiser currently active, which has raised over 45,000 more, because we're hoping to have the funds if and when the mouse trials prove to be effective so that we can move to the next stage quickly. There is the pigacdg.org website that I mentioned, and an active PIGA Facebook group. And we are working with Dr. Chow's team on both the medium throughput screening and building a patient registry for PIGA. And last year, we also started Emmett's Legacy Fund, which I hope everyone has heard about, and everyone with a CDG or with NGLY1 is eligible. This year's application will be out in the next few days, so keep an eye out for that if there's equipment you've had trouble getting that your child could use. And finally, I wanted to close out by saying this. We know that the rare disease life is extremely isolating. Reach out to other families as much as you can. I know we found so much inspiration and motivation by talking to other families. Attend things like this conference. This was us in 2018, or get to know other families in your area. Thanks to social media, we are able to keep in touch with people in different cities, states, even countries. So make sure you are not alone in this journey. Thank you again for having us. Please feel free to reach out to Steve or I at any time.